Okay. Thank you so much for the invitation. I really enjoy and like when I'm invited by students, because it's really people who are interested, that they come not because they're forced, but because you like to, and it's really exciting for me to be here. I know that the audience is quite diverse, so I'll try to give an overview of what we do, and if you have any questions, please ask during the talk. I want to make sure that, I'll try to talk about some more machine learning stuff and more applied stuff. So first, before I start, I want to tell you about myself. As was said, I come from Italy. I was born and raised in Florence, and I studied I did my bachelor degree in physics from the University of Florence. And then I moved to Trieste, where I did my master and PhD at the International School for Advanced Study. That is a theoretical physics school. It's a little bit farther north than Florence, but still in Italy. And then after that, I decided I wanted to stay in science, and I moved across the ocean and went to San Diego, where I was for 3 years a researcher, postdoctoral researcher. It was a beautiful time, and San Diego is amazing, a beautiful ocean. And after that, I moved to Houston as a professor. But Houston is not as nice as San Diego, but the science was really exciting. I stayed for almost 20 years, was professor of chemistry, and 4 years ago, I moved back to Germany, moved back to Europe in Germany, and I'm now Einstein Professor in the Department of Physics. So this is about my science, but I also do other things besides science. When I don't work, I like to do underwater photography. This is me with my camera underwater. Of course, this is not in Berlin. It's in some warmer place. And I like to take pictures of sharks mostly, like this, or very small things. Like this is a few millimeters. And it's with the macro lens. I like to take this kind of pictures. So this is about me. And let's start trying to explain why I do what I do, the philosophy of our approach. So molecular science is about nanoscale, mostly behavior. And if you think about when you study just a small molecule like water, a few atoms, what do? We try to have the most accurate representation. We want to study the probability of the electrons around this molecule. We go to the very, very small and very sophisticated theoretical methods, first principle, to try to understand everything, the fundamental physics, because that gives us the bonding properties, the whole chemistry, the reactivity, everything. If we have the same thing, water, but instead of one molecule, we have an infinite number of molecules, is what we call the bulk, we are not interested in the same thing. We don't care about the position or the probability of the electrons around each single molecule. We care about what we call the phase diagram. That is the thermodynamics. We care about global properties, the behavior of the overall system. So it's the same molecule. Here is just one, and here is an infinite number. These are the two extremes, the two limits, and we change the variables. We change our interest depending on the number from 1 to infinity. The problem is usually, in reality, we are in between. If we are a nanoscale object, there are large clusters that is neither 1, but is not infinite. Is a finite number. It's not Avogadro number either. It's like 1,000, millions. That is quite far from infinity. We have heterogeneity. We have interfaces. So it's a very messy reality. So it's a fair question to ask, what are we interested in here? Because most I mean, if you don't know, you can take this approach and try to solve the most accurate of everything. But then, is this appropriate? Because we don't know. I mean, maybe we're interested in global properties and we don't know what are the correct variables. What is the appropriate resolution? So these are questions that we try to answer using physics and machine learning. So our point of view is that we want to understand what is the minimal model, the minimal representation of the system that gives us reality, the minimal model that reproduces the relevant physics. And we have evidence that we don't need to go as accurate as here. We can disregard the electrons, we can disregard some of the atoms, and we can make effective models at intermediate resolution. So, and this is not just to speed up simulation. Clearly, you can already see that if I put all the details and I try to model everything starting from first principle, that in physics would be the Schrödinger equation, you don't go far. You can solve the Schrödinger equation for an hydrogen atom, and then you have to approximate, and it's really complicated, and for large molecules, you forget it. So, we cannot do this in practice, but it's not just speeding up simulation, it's also generating understanding, because we want to understand what's important and what's not important here. So this is what we call coarse graining. So we can start from this limit, that is the first principle limit, where everything is in the system. We want to characterize every single degree of freedom, every single water molecule around. This is a small protein around the protein. And we know this energy function. This is the energy of interaction of all the molecules is given by physics, by first principle. If we start to remove, for instance, we can remove the water. We say, I want to model this without the water. Or I can say, I want to, instead of putting all the atoms, use some representative atoms and less. Then what is the energy function I should use? I don't know, because I don't have first principle anymore. I lose first principle when I start integrating out degrees of freedom. So we need to find this energy function. And this has been a problem in coarse graining for a long time. This idea has been around in physics. This idea has been around forever. Physics is all about making the simplest description of reality. But in practice, if you have a large molecule, this doesn't work as easily, because the mathematics behind this is very complicated. I'll give you a glimpse of the mathematics. So to formulate this problem mathematically, you can take different approaches. You can say, I want to go bottom up, meaning I want to take the fundamental description, the fine-grained description where everything is in it, and rigorously integrate out degrees of freedom. And I'll show you the math for this. And then I'll get the equivalent model with less degrees of freedom. Or I can go top-down, saying, I don't care about how I go from first principle. I just want to reproduce experimental data. And then this is an inverse learning approach. And you can do it. And there are examples of this. Actually, I have to say they don't work very well, but people still use it. Or you can try to say just, I want to reproduce some structures. And there are other approaches also, they don't work very well. What we think is the most fundamental approach, the most rigorous approach, is to go from here up. And this has been limited for a long time because of the tools that were available. Now with machine learning, there is a kind of revolution in this field. So the math is the following. I have an energy function here that is given by first principle. I integrate out some dimension. I say, these atoms, I don't care. So instead of a region in this high-dimensional space, I go into a point in a lower dimensional space. I integrate out some direction. I have a distribution in this space that is given by, in physics, we call it the Boltzmann distribution, but it doesn't matter. In machine learning, you can say you have a probability distribution of a molecule being in a certain shape. It's a probability distribution in very high-dimensional shape. I want to preserve this distribution in the low-dimensional space. So I want these two distributions to match. So I have a projection operator, a map, the matrix basically, that tells you how you go from this high-dimensional space to here is dimensionality reduction. And you want to find the potential, the energy function that preserves this distribution. And you already see, talking about a dimensional distribution, it's easy to put machine learning into the mix. So this is the equation that tells you, if you could solve it, how to get this energy function. Preserving the distribution means that this energy function is given by the marginal distribution, the logarithm of the marginal distribution of the original distribution. So this is, if you want to preserve the distribution, this is what you need to do. This tells you how you go from this energy function starting from the first principle energy function. So in principle, you know, but in practice, it is impossible to solve. This integral is like millions of dimensions. You don't solve it, not even numerically. So this is how it works in 2D. If I have this energy function, this is color-coded, the blue is the minimum. I say I'm not interested in the second dimension because it's like just boring. I'm interested in the x direction. So I integrate out the y and I find this in x. Makes sense. I find the two minima and then I just focus on the x direction. And these two potentials have the same distribution. This is 1D, this is in 2D, but they have the same relative probability of being in one point or the other in an x state. We want to do this for millions of dimensions. So this is the way to do it. We can reformulate the problem, and there's been quite a bit of work on this, as a machine learning problem. You can reformulate the problem as the minimization of a loss function, where, let me go through the ingredients here. This F is a force. Force is a gradient of a potential in physics. So this is the force. These are your labels. These are the forces, but is in the fine-grained space. You have an example of your system. Somehow you have instances of different configurations of your system spanning this probability distribution with forces, with force labels for each atoms. You project, this map is the one that goes from the high-dimensional to the low-dimensional space, because these are high-dimensional forces. You project them into the low-dimensional space, and you compare with the force of the coarse-grained model. That is what you want to learn such that the gradient of U, the difference between the force minus this gradient is a force. The difference between the projected force and the force of the model, on average, is minimized. Now, this has been proposed in 2005 and has not been used much in practice because this loss should be minimized in the whole functional space. The function U that minimizes this loss is anywhere in the space of integrable functions. So if people use it variationally, they try to propose an ansatz for you and minimize the parameters, and then you may be very far from the real energy function, because U can be anywhere and can be a very complicated function. You have no clue what this U is. And so this is the perfect role of machine learning, because machine learning allows to go with the neural network. You can approximate, in principle, any function if you have the right architecture and the right number of parameters. So this is what we did. We proposed in 2019. And before to use it, I need to tell you something about these laws. So first of all, so this is what we proposed. We said we use these laws, and the neural network, instead of an answer for the function, we plug a neural network here. And this is usually a graph neural network. We use, I talk about GNet, that some of you know, has been developed in Klaus Robert Mueller group and other similar graph neural networks. We propose to divide this energy function in a part that we call prior, that we can guess from physics, and the correction is the network that tells you, adds what you don't know. In particular, it adds all the many-body interactions, all the terms of all the atoms interacting with the other atoms. You cannot write a general functional form for that. Then the sum of these two gives you the energy. You take the gradient, you get the force, and you plug it in the loss. This is the force, you plug in the loss, and you minimize it. This is what these are the results we can get with the state-of-the-art methods we have developed along the way. So this is what we want to reproduce. This is the ground truth probability distribution. These are two important variables, collective variables that span the probability distribution. Here, low means high probability, because this is minus the log of the probability. That is the energy. And so you see that if this is the probability of visiting different modes of this distribution that correspond to the states of a protein. One is unfolded, is very disordered, that is this mode here. Two is folded, but not in a good way, and it's misfolded. And three is correctly folded, that is here. If you have this protein in reality, it can go from one of these states into another, and this is the distribution. So this is the reference, is what we want, and this is what our model trained on the data reproduces. And you can see they are very similar. We can be more quantitative and project take just one of these coordinates instead of both, and this is the one-dimensional distribution, and you see they match quite closely. In particular, the error is mostly on the where we have less data, where the distribution, the top of the barrier here is the minimum in the probability, because remember is minus the log of the probability. So these are where is more data scarce. So it makes sense that there is an error. How do we do this? How do we learn this? I told you about this loss function, but there are certain things that we need to do on the loss function. So this loss function, this learning problem is super noisy. It's very data inefficient, because this loss function never goes to zero. The minimum, there is a minimum that corresponds to the correct U, but the minimum is pretty high, because this is in a different dimensional space. This F is very adimensional. So for any low dimensional configuration, there are many high-dimensional configurations that correspond. So you have a distribution of forces for each U. And that means that you have a large noise. It is cannot go to zero. But so this is the mathematical formulation, is an ensemble average, but in practice we do a data-driven approximation from the data we have. We can do some mathematical trick and write this loss as a function of three components. One is the noise, what we call the noise, that is this base, that is the minimum of this loss function that you don't know, is what you would get if you have the exact U coming from the integration of the degrees of freedom. Then you have the variance of the parameter variance, and the bias as usually, and there's always a trade-off between bias and variance. But there is, instead of going to zero, the loss as this baseline, this noise that is pretty high. And so makes the learning problem very different. But if you notice here, in both the bias and the variance, there is this U, that is the learned function, the neural network. Here, in the noise, there is no U. So this does not depend on the parameter of the neural network. It's something that depends only on the dimensionality reduction. So we can try to optimize this, because it turns out that this projection here, you have some freedom in this projection. You can pick the way you do the projection. So we can optimize it to try to reduce, to bring down this noise as much as possible. And then we do the optimization, trying to find a trade-off between bias and variance. But first, we can optimize the noise separately. So it's a double learning problem. First, you try to optimize the noise, and this doesn't involve you at all. Then when you have tried to take down this noise, you have an easier problem in learning you. This is what we do in practice. What it means is that even if your dimensionality reduction says, for instance, this is a molecule, I take only one atom to represent this part of the molecule. This is part of a protein, is an amino acid. I take only this atom here, and I throw away everything else. For the force, I can take a contribution of all the atoms. I can do a different projection, and this reduces the noise dramatically and makes the learning problem much easier. In practice, let's go here. In practice, this is what you want to reproduce. This is what I showed you, what we can do if you optimize the noise. And this is what you get if you don't optimize it. And for other proteins, it's even more dramatic. This is the distribution. This is the optimized learning problem. And this is what you get with the non-optimized projection. So this is important. When we discovered this, we made a big step forward. Another thing is we've done recently, we have submitted it last week. We realized that there is a nice parallel between this learning problem and the very popular now the noise in diffusion models. Because what are we doing here? We can rewrite this, what we call force matching loss, like this. This is the force coming from our energy, what we learn. This is the derivative, the gradient of the neural network. The gradient with respect to the variables R, that are the corresponding variables. This is the label, this F, that is the I-dimensional force, and this T is this projection from high dimension to low dimensional space. Now, if you consider what the denoising score matching loss is, that is the part of the denoising diffusion model, is very similar. This is what you can call the noise force. So what you do, you have a distribution R. You noise this distribution with the conditional density. Given the position R, a point R in this dimensional space, I perturb it to find R prime. I can associate to this a force. I take minus the log of this distribution, and I find the force. After minus the log gives you the energy, and then I take the gradient, I find the force. Just applying the same definition. Here I have these labels correspond to a distribution. I have an energy, I have exponential of minus the energy is the distribution. I take the gradient of the energy, I have the force. So I apply the same prescription. And this is the neural network. This is the denoising neural network that you learn. And then you do an average in a denoising score matching loss. You do an average over the noise. And you do it in a denoising diffusion model, you do it for many different noise levels. Let's focus on one noise level, small. What you learn is this S. The only difference between these two, this is a force, this is a force. This is what you learn, this is what you learn. The only difference is this average over the noise. But maybe I can combine the two things because they are so similar. I can introduce a noise and average over the noise. And this is what we try. So this is our atoms. We introduce a noise. So we move the atoms a little bit. Then we combine with these atoms have forces. These are the labels. We combine the force of restoring force of the noise, that is the denoising, with the force we have, and we get another force. It's just a vectorial combination. And then we do the coarse graining. We cancel the atoms and we take some representative atoms. And this works much, much better than either one. Because I could use a denoising diffusion model. What I'm trying to learn here is a distribution, right? So instead of using the forces, I could say I use a denoising diffusion model to recover this distribution. This is also not working very well. This is a super-dimensional distribution. And also the problem is that the reference usually is not really the distribution you want. It's an approximation because it's a sample of data and it's very difficult to sample. So maybe the data distribution is not the distribution you want. So this is why we use forces. But if you have a good distribution to start with, a good reference distribution, you could use the denoising diffusion model. And people have done it. Many people use the denoising diffusion model to trying to find protein models. But they are very expensive and they require a lot of data. And also this problem here, the force matching loss that I have is very expensive and requires a lot of data. Turns out if we do this trick and combining the two ideas, so I have the force labels, I combine it with the denoising labels, the storing denoising force, and I do the coarse graining, it learns much better. And then I do average over differentialization of the noise. I take, for instance, a Gaussian variate for the noise. It's much better. The idea is, if I have the forces only, I'm trying to recover the energy from the forces. I'm integrating the forces to find the energy because the forces are the gradient of the energy. If I have the noise only, I minimize the difference between the distribution. I try to recover only from the distributional information. I don't use the forces. I just try to match the distribution. But if I have the forces and the noise, I can learn from both. I can learn from the forces, and these are independent information. I use the distribution of the data, and I use the labels I have on the data, and I get a better learning problem. So this is the way we do it in practice. So you see, it's very similar to this one, but now this force comes from our labels. And what this T does combines the forces from your labels, the molecular dynamics forces, with the noise forces that come from this noise, corruption of the data. And here, while I do this, I also do a coarse graining. I go from the high-dimensional coordinate to the low-dimensional coordinate. So I also throw away some stuff. And this works much better. So this is a bit busy graph. So I'm showing if I use only the noise, that is basically a denoising diffusion model to learn the distribution. The ground truth is this thick gray line. If I have a lot of data, this is 100% of the data, I have millions of data points in the training set. I do an okay job with learning only the forces, only the noise, or both. This is the error I make. Okay, using both is a better, error is closer to zero, but it's okay. But if I start to use less and less data, if I use only 2% of the data, so it becomes much more efficient, I still have a very low error if I use both denoising and forces, while the error diverges if I have only denoising or only forces. In particular, I learned the distribution doesn't have anything to do with reality because these two modes, these two minima in the distribution correspond to completely different states. This one is the folded state. This one is the disordered state. And you see, if I use only the forces, I learn only one mode, only the unfolded. And this is the difficult state to learn. And this is also the error I made here with only the denoising is pretty large. While if I use both, I get it very well. So we are very happy about this. And you see here, introducing noise reduces the error dramatically. Even with 1% of the data or 2% of the data, I have an error that is acceptable. 1 kilocalorie per mole, 1 kBT of error is completely acceptable. So what do we do with all of this? We have worked a lot on the technique, the architecture. Now we want to use it in practice. So we try to learn what we want to do. We want to learn one model that is universal, one model that can give you the probability of any protein, not just the ones you have used for the training. It's like, I guess you know AlphaFold. AlphaFold has been trained on the part of the PDB, the protein data bank, and then you give it another sequence and gives you the structure. Same thing here, but for the full distribution, not just one structure. We want the full distribution that you get doing, letting the protein move, not just frozen in one structure. So if we first did it we did the limited test, we learned a model that can generalize to 12 proteins. This model can generalize to learn the distribution of 12 proteins. But recently, we have put together a model that is universal, it's called transferable, that can generalize to, in principle, any protein. We still have some work to do, but the proof of principle works quite well. The way it works, this was many years of work. We had to create very carefully the dataset, the training dataset, to have diverse sequences and diverse structures. We had to work very carefully on the physics constraint we put into the network, because if you put the physics constraints that are incompatible with the learning problem, it just explodes. The architecture is something we're still working on. What I'll show you, we use SNET that has been proposed for other problems by Klaus Robert Müller group, but we are inventing other architecture, mostly because of the locality of SNET. That is a bit of a problem. We are trying to play with ideas to extend the range of the interactions. And then the validation is also very important, because eventually we want to compare with reality, with experiments. So the data set we created, and for this experiment I'm showing you, is just 50 proteins, a little small. We ran some simulation with all the bell and whistle. So these are very expensive simulations. It took months to create a data set to try to sample different configurations from the distribution corresponding to each of these proteins. And then we have fragments of proteins coming together. And then this is the validation set. So these are real proteins that were not in the training set. Not only were not in the training set, but were very different. They have very little overlap with the training set. If you look at stretches of the sequence and you compare it with the sequences in the training set, the overlap is very small. And they are longer than the proteins in the training set. The proteins of the training set are 50 residue. They're like chains of 50 elements. And here we go up to almost 100. So it's really extrapolation out of sample. And it works. This is one example. This is the protein I showed at the beginning that was not in the training set. This is the ground truth. The important thing here is to find the 3 modes of the distribution. And indeed, we find the 3 modes of the distribution. This is the deepest, that is the folded state, is the most probable state by far, and we get that. And then there is the unfolded, and then there is the misfolded. This is a bit smoother than this, and this we expect this for some physics arguments. We expect this to be smoother when we learn an universal distribution, and it works for many proteins. We compare with ground truth for different proteins for which we can afford the simulation with all the details, and we compare with different approaches that people have proposed in the past that don't work very well. This, we can then do some prediction. We can take proteins from the PDB, for which we have the crystal structure, and we can try to build the distribution. We don't know the distribution because you cannot use the existing methods to sample it because it's too expensive. So these are our predicted distributions of this protein, but the most important thing is that the lowest minimum, the most probable state, is the crystal structure. We recover that. So that's interesting. But also we can find alternative modes that are disordered. And this you don't get with the existing methods, because to go from here to here with an existing, with the state-of-the-art molecular dynamic simulation will take too long, like years. We cannot do it. But with our method, we can, because this is a very simplified representation, we can do it in a few hours. This to show you how it works. This is another protein, the validation set. We take the protein, the initial configuration, we take it completely extended, and we let it go. This is done by integrating the neural network. Every step is a call of the neural network that gives you the forces and defines its final state, that is the crystal structure. But it's not just one state. You see, there are excursions from the field crystal structure that are very important for the function of this protein. The minimum energy, the most probable state, is the crystal structure, but if it moves around. These are other examples. This is another protein that moves around the crystal structure, and we can do it for many proteins. We can compare with fluctuation around the crystal structure obtained with other methods, and you see it's again smoother than the reference, but we get the correct modes. Then we can do something that really you cannot do with other methods, that is intrinsically disordered proteins. So there are certain proteins that do not have a crystal structure. You cannot find a crystal structure because they just don't have one structure. They are disordered. So the distribution is different. It doesn't have a deep minimum. There are different modes, and they're all very shallow. So we use our method with this, and we have experimental data. We have NMR structures. And if you see underneath here, this is our distribution that we predict with our method. So underneath here, there is a shallow, a very broad minimum that is a region of high probability, and all the NMR structure are in this region. But then we have also another region with much lower probability. This is the difference of, maybe a few order of magnitude difference in probability. And this cannot be observed experimentally because the probability is too low. But we have evidence this is real. We are collaborating with experimentalists, and also we have tested it with other methods. And this is what we predict. This is a disordered ensemble, but somewhat compact, more compact than these other structures that are predicted by NMR. So this, you can imagine, we have a lot of collaboration with experimentalists to try to use these methods to understand something on protein that you cannot study otherwise. I want to tell you want to switch gear and tell you something else that we are doing. Well, besides developing these methods for application on protein study, we're also trying to understand what we learn. And this is a collaboration with Klaus Robert Müller here at TU. Klaus has developed these interpretable AI methods, in particular this layer-wise relevance propagation. And talking with him, we discovered that this layer-wise relevance propagation is what in physics we call multi-body decomposition. You just try to find the relevance, the contribution of different subsets of the graph. This is the graph neural network. You try to find different subgraphs. We want to find the relevance of different subgraphs to the final output that in our case is the energy. So we decompose the energy into contribution of all the different possible combination of part of the input. This is the way it works. Basically, you do look at, this is the graph for this fictitious molecule. The neural network goes from the graph into an energy function. And then this walks back from the output to the input and tries to see, for instance, here, if the output is, if you have a given output, what is the contribution of this subgraph? You walk back through the layer in what we call the walks, and you look at the connection of the nodes of the graph to another nodes of the graph layer by layer. It can connect to itself from one layer to the next. And so we can define all these in this case, this two-body contribution is a set of, like this two-body contribution is all these subgraphs that connect these two nodes. We group them together, we call it two-body contribution. So we decompose the output into contribution corresponding to different inputs, and for us the inputs are the atoms. So these are groups of atoms, how the groups of atoms together, the interaction of groups of atoms together correspond to the energy. We first did it for simple system like water. We coarse-grained the water from, this is the water molecule, we just consider one atom to represent it. And then we decompose the energy into this multi-body decomposition. And you can do the same for methane. And we use two different architectures, both from Klaus Robert group, PAIN and SO3net. These are very different architecture. They have different features, but they give you the same interpretation. So this was very interesting because it means that they learn the same thing. And in fact, the energy should be one. There are different ways of learning the same energy, different way to represent the same energy. In fact, this is the two-body contribution. And you see there is a minimum corresponding to the peak of the distribution of nearest neighbor. This is called a radial distribution function. This is the correspond to the hydrogen bonding in water. And it gives you a minimum energy correctly in both models at the same place. Methane doesn't have hydrogen bonding, so you have only a repulsive core, and both models give you the same thing. We can look at the three-body contribution, and if you focus on the continuous red line, you see that there is a minimum in the same place that corresponds to the tetrahedral arrangement of water, while for methane, there is no three-body contribution. In fact, and this is for both models, in fact, this tells you that you can learn, you can do a regression for methane with splines, with two bodies only splines, and it works. In fact, we tried, it works. This tells you that there is no three-body contribution in methane, while for water, it's very important. You can do a regression with splines for water. Then we applied it to our protein models. We applied it to this protein here. This is, we use PAIN, which is what we obtain for this is the distribution we get. And then we look at the attribution of all the possible contacts, interaction between the different pairs. So this is the contact path. We look at each point here is the interaction between one residue along the sequence and another residue along the sequence. And if we look at the folded state, all the contribution, this attractive means important relevance. So negative relevance is good, is important contribution. And you see all the important relevance, these highlighted regions are the secondary structure of the protein, the contact formed in the protein in the native state. And not surprisingly, all the important contribution are where in the contact they stabilize the folded state. While if you go into the folded state, you have a lot of destabilizing and they're all over the place. Then we can do mutation. These are the most important two-body contributions, and you see they stick together different parts of the folded state. And these are the three-body contributions. So you have a network of 2 and 3 body that keeps the protein together. But we can do mutation. We can say, okay, now I mutate this into another residue, and you can see the relevance propagate, how the relevance changes when you change the residue. And you can see, for instance, this is the interactions you disrupt if you mutate this one. These are the interactions you disrupt if you mutate this one. So it really gives us a way to read what the neural network is learning and interpret it in terms of what the biologists want to know. What are the important interactions? What happens if I mutate one side? How does the energy change? How much time do I have? Okay, so I skip all of this. I want to tell you briefly the inverse problem. So people tell me that, okay, you show that you can reproduce a distribution, but the distribution was created by a model to start with, was a simulation. What if you want to reproduce reality? What if you want to reproduce experimental measurement? Because your ground truth may be an approximation itself. So this is, in physics, we call it inverse problem, because you want to get the detailed model, the model from observables. So we came up with the maximum likelihood approach to do that. The idea is to write the likelihood as a function of the model parameters that we can do by some statistical mechanics trick. So these are the labels you have. These F are the experimental data. E is the prediction of your model. That is the function of the parameters of the model. And you write the likelihood like an error model. And then you maximize this likelihood. So you minimize minus the log. The minus log likelihood is your loss function, and you use the usual tricks to minimize it. So this must be done iteratively because this is obtained with perturbation theory, so you cannot take very large step in parameter space. So there is some regularization in this loss as well. So with this, we can test different functional form to see, for instance, this is our energy. This is the physics part. And this is what we don't know, what usually we put a neural network in. We said, okay, instead of putting a neural network, what error do we make if we use simple function like two-body only splines? And to make a long story short, it doesn't work with splines. You need to put multi-body terms. So here, instead of a neural network, we put some more complicated function that takes into account the surrounding of the residue. And you see that there is, if you use only two-body splines, then you cannot recover experimental data. So these are experimental energy differences upon mutation, and you cannot, the experimental data are on this axis. So if you do two-body only splines, you're very far, especially for this one, you're very far from the diagonal. But if you use many body, like a neural network, this is not a neural network, this is simpler, but you go much closer. And from this, you can understand, if you look at where these mutations are exposed to the solvent, and it means that in order to take into account of the solvent, you need to have many body interactions, like a neural network. Now we are incorporating everything together. And now our ultimate goal is to combine this with the learning of the distribution from simulations to have not only something that can reproduce the distribution, but also experimental data. With this, I would like to tell you that I'm hiring PhD, postdoc, master at any level. So if you're interested, contact me. And I would like to thank all the people who have who work in my group and the funding agencies and all of you for your attention. Thank you.
Awesome. Thank you so much. If you have any questions for Professor Clementi, there's going to be a microphone. And Anna has some.
Hey, thanks for the talk. That was super interesting. And there were so many results in there. I just wanted to know what is like the next question you want to ask to your approach? Like, do you have something in mind?
Yeah.
What your next goal is? Yeah.
So the most important thing we are working on right now, I think, is the long range interaction. So if you know, graph neural networks are local. They have a cutoff. They see each node sees a certain number of nodes nearby, and then you extend the field of view with the message passing. But you cannot go infinitely long. The problem is that in physics, there are interactions that cannot be cut. Like if an interaction between two beads, two atoms, goes to decays to zero at infinity, slower than 1 over r cubed, you cannot cut it. If you cut it, you'll make an error that it diverges. So this is a problem in all the message passing neural network because they're all local. They start from a node and go farther and farther, but it is not scalable because if you put everything in it, it scales like n square, and it cannot go to these huge systems. If you use a transformer, it scales like an n square. Here we have system. We want to go to system with at least thousands of degrees of freedom. So you cannot scale, you cannot simulate it in a reasonable time. You're back at the problem at the beginning that it takes too long. So we're working on this. We tried different approaches. One way that works, but is very expensive, is to go into reciprocal space. So instead of putting a cutoff in distance, you put a cutoff in frequencies. That means that you go very far, but it's very slow as well. So we're trying different things. We're playing with, we try to use physics idea like mean field and things like this and put it and transform it into an architecture.
Thank you very much. It was a very interesting talk. A very basic question, maybe. I think I read it between the lines, but how do you generate your training data? Is this like very expensive?
It's super expensive. So we run simulations to generate. So for the next generation model, what I showed you is a paper that is now in revision in Nature Chemistry, and it's been submitted last fall. In the meantime, we have been generating the next generation data. It took us 1 year to generate the data, and it's still not enough. So this is why we are playing all these tricks to try to lower the data demand, because we have a lot of GPUs, fortunately. We have access to many GPUs, and we use them all.
Okay, thank you very much for your talk. I was wondering, so just to basically check if my intuition was okay on this. So you do force matching, but then you also introduced the idea of adding noise to it to basically benefit from like the
From
diffusion
both.
process, but also from the force matching.
Yeah.
I was wondering if one could see it also as like a diffusion process where the forces are like conditions.
Yeah.
Yeah?
Yeah,
Okay.
I think so.
Thank you.
And actually, we are trying to understand if we can go on the other way around too, if instead of helping the diffusion model helping the force matching, maybe the force matching can help the diffusion model. So I don't know. I'm not. I know my field, but I don't know other field of machine learning. Maybe it can be used by other machine learners.
Okay, thank you very much.
Thanks. Maybe a simple question. So how large are your current models? So in numbers of parameters?
I don't know, but let me think. We have 64. The Fisher space is 64. We have about 4 layers of continuous filter convolution with about 20, 30 radial basis function. So, and then we have a multi-layer perceptron at the end with the 2 layers. So it's not huge. Fisher space is 64, but with 4 layers, not huge, but big enough.
Yeah, first of all, thanks for this informative talk. And my question is, you showed us how proficient your model is at, approximating these protein structures. And my question was, from this current point in your research, how beneficial is this for other areas, for example, like medicine? Like, can they already use, or do they already benefit from your results?
So one thing we are doing in collaboration with some doctors at Charité and some other researchers at the FU is to try to use this for trying to understand the immune recognition. So if you know how the immune system works, there is the adaptive immune system that basically is, there are some proteins that bind to peptides that come from can be either already in our body, like a self-peptide, or can come from a virus, a bacteria, something else. And this protein somehow recognize binding or with different strength, recognize if the peptide is self, and if it's self, they don't do anything. If it's outside, it's foreign, they activate the immune system. This is still not understood, because the dynamics is very important. It's not just lock and key. It's not just binding. The dynamics around this state is very important. So the doctors came to us and asked us if we can predict, if they give us a sequence, if we can predict when you change the sequence, if it still binds or not, because that's the first step for the activation of the immune system. So this is what we want to do. I wrote a grant for that, so if we get it, we'll start doing it. And other things, we have other collaboration with these intrinsically disordered protein are very important, because if they aggregate, they cause disease, and they are very difficult to study experimentally because they don't have one structure, and in simulation takes forever. So we have several collaborations to try to do that as well.
Okay, I have 2 more questions, and then you can ask afterwards, maybe also.
So just a 2-part question. One is in terms of the practical speed-ups. What are the speed-ups that you've got versus your benchmarks? And secondly, in terms of the implementational details, is there a specific computational step that, are you making it off the shelf? Or are you using off the shelf, computational details? Or is there a bottleneck which you've identified, which if you make it faster, that would make it, an order of magnitude faster, like flash attention versus attention, etc.?
So all the science I showed you is with software that we wrote ourselves, and we are physicists, we are not computer scientists. So we get the speed up of a couple of order of magnitude, but it's still not good enough because we want to scale it up. So now we got some help from computer scientists at the Supercomputer Center in Munich, and they want to do our software professionally. The main problem is that we are using PyTorch and it's slow. So we want to do everything in CUDA. When we have a final model, we want to put it in CUDA, and we want to do it in a modular way so that we can speed it up significantly. I don't know how to do that, so they need to do it.
Thank you.
There was one slide where you showed a training process of, like a visualized training process of a protein. If you could go back to that. I don't know if you remember which one I mean. Basically the residuals.
This one? Oh.
No, a lot further back. It was still in the generative part. This one is story. Go further. Further. This one, I think it was a video.
This is not a training. This is after the training, what we do with it. The model is already learned. This is the dynamics. This is time.
Okay.
This is how a protein moves. Each step is the neural network giving an energy. So it integrates the dynamics using Newton's laws of motion that are the forces given by the neural network.
Okay, understood. And what's the black thing?
Black is the crystal structure.
And we're converging to the crystal structure.
Yeah.
Okay.
It finds the most probable state as the crystal structure that it should be, but also it gives you the full dynamics.
Okay.
The full distribution. So if you put this in AlphaFold, it gives you 1 point. The difference in what we do is that we get the full distribution.
And you just, as a starting point, you just sampled randomly from your distribution? Or what's the start?
No, the starting point is 1 point that is very far here, that is completely distended. It's completely extended chain. And then you let it go, and according to the laws of physics, it goes to the minimum energy. So this is where it goes.
Okay. Thank you very much. Then I understand this slide better.
Thank you.
All right. Thank you so much.
Thank you. Thank you very much.